Hi everyone, I'm Hadi T from CRC, Department of Biomedical Engineering. I'm going to tell you about crystal ribcage and its application in cancer, not aging. Aging is also another application from the previous research on tap. So we are interested in, imaging the lung in real time at the cellular resolution and in a functioning lung. To do so, basically, we want to use optical microscopy. We take the lung outside the body of the mouse here, and the lung is ventilated, the lung is perfused, and then we design our own ribcage that has the same properties and geometry of the actual ribcage, but transparent. So we call it crystal ribcage, and it allows basically any optical imaging modality. So, these were supposed to be videos, but here I don't think I can run it, but let me try quickly. No. So these are the videos. Here you could see the lung basically could function inside crystal ribcage. And here was another video. You could see the alveoli are moving around. And this is the real-time basically function of lung, the respiratory function of the lung at the alveolus resolution. You can basically zoom in further in. You can see the basically melanoma lung metastasis here in green. The CD8 positive T cells are in cyan. This was another movie where you could see basically the T cells are moving around. And this is immunogenic version, another melanoma model. You could see basically a huge difference between the immunogenic versus the non-immunogenic model in terms of the T cell motility and their infiltration and the dynamic basically behavior of T cells inside and around the tumor. So we could demonstrate how crystal ribcage is useful to do a multi-scale analysis of a functioning lung. And this is basically a truly multi-scale analysis from whole organ down to cellular resolution. We could basically demonstrate this for healthy lung, for lung metastasis, primary lung tumor, bacteria, pneumonia, fibrosis, and emphysema. Basically any lung disease that would affect the lung parenchyma. So we recently tried to connect the circulation of an awake mouse to the circulation of this lung inside the crystal ribcage. So this mouse is awake, so we are not limited to the anesthesia time. And in real time, it donates basically the blood to this lung. First, this would basically make the maintenance of this lung inside crystal ribcage much longer. But also, there's a lot of interesting study we can do by mismatching this mouse and this lung. This was another video. I wanted to show that the mouse is happy. It's walking around. In real time, the blood is going through tubes into crystal ribcage, which is up here. In the video, I could show you there. And the blood comes back to the mouse. So it's basically a closed loop. And this is another video that I can't run it here. This is the chip we put in between. The blood goes in and comes out from the lung. We can in real time see all the circulating cells. So this is basically an in vivo cytometry. We can count the immune cells, circulatory T cells, and you could also see the hemodynamics of the basically lung capillary in real time. So how can we study cancer with this model? There are so many studies we can do because of the dynamic, basically, capability of the system, but also because of this mismatch. We can do a lot of mechanistic study. One of the questions we want to answer is if we have immune checkpoint blockade. Would it stimulate the resident CD8 positive T cells, or would it stimulate the circulatory CD8 positive T cells? We can pick a mouse with, let's say, green T cells. We can pick another mouse with red T cells, and then we can stimulate with the ICB, and then we can see the basically circulatory and resident cells at the same time inside the lung. And we can study the differential effect of ICB on resident versus circulatory T cells. There are, again, so many applications. I'm happy to talk to you. And with this, I would like to stop and thank you for your attention.